Hello, this is Robert Faust. I'm out here in my farm area in near McMinnville, Oregon. And I'm just trying to do this video outside here. See, it's a nice day, blue skies here in the Pacific Northwest, and very blue sky. So, September 18th. But I've made an important breakthrough, and I thought I'd just do a preliminary introduction to the concept. Now, we managed to get a license from USDA for a piece of technology that they developed in the Agricultural Research Service of the USDA. It's a fungi developed and found it by accident, and doing tissue culture, plant tissue culture. So there's a contaminant on a Petri dish. So that Petri dish, the next Monday, after they went home for the weekend, the microplants, the plantlets, were remarkably larger and more vigorous, where they found these spots, contaminants on the agar plate, on the Petri dish. It's a black or dark colored fungi, and it has a high melanin content. So since then, they patented this, and they licensed it to me, or Fausts Bioag, and we've been working on it for about 4 years. And it's remarkable. And the effects. Now, just finished a recent research paper where we exposed cannabis seedlings to 14 to 21 days exposure to the VOCs, or volatile organic compounds, produced by this fungi in a special canister or device that I developed to release, to maintain the culture within an enclosed container, and that selectively emits the VOCs. So what we found out, now we have new technologies available to us we didn't have before, and that's to be able to test, essentially every terpene that we know about in cannabis, and that's a couple dozen potential terpenes. Okay, so what we found in the terpenes was that where we exposed the plants, identical plants, control and treated were exposed to terpenes in the seedling stage in a humidity dome or a tiny mini greenhouse. And the controls were just grown a normal way. And then these plants were grown with hydroponic media, just plain old, a Hoagland solution, NPK. We did add fulvic acid, however. But anyway, so the difference was probably a double yield. The yield was about double. And then testing, it went from 8.5 or something like 8% THC to 12% THC, and almost 40% yield increase on top of that. Okay, well, then the terpenes expressed treated with the VOCs, there was terpenes detected that were not even detected in the control. That's the remarkable part. In those particular terpenes, which I get into real detail about this in some things I've written, they are partially, the terpenes that we found in the VOC that were missing from the control are ones that have to do with resistance to diseases like, let's say, powdery mildew, a whole range of other plant diseases that provides it, essentially triggers resistance in plants is what's happening on a molecular basis, on a cellular basis, turns on the plant's natural mechanisms to protect itself against pathogens. That's fungi, bacteria, it can be viruses. Plants have it all, right, in terms of plant diseases. And that's the kind of work I did for years. what's causing decline. And fungi are a two-edged sword. They can cut two ways, There's good ones and there's bad ones, and the good ones can protect the root system from bad ones. We have mycorrhizal fungi like our VAM product. Now this is something that activates the plant's protection and plant growth regulator / biostimulant production within the cell of the plant itself. This is not external. It's not adding a fertilizer. It's not adding a hormone. It's not adding a biostimulant. It's doing all the things that biostimulants do, including the products that bioag sells, like humates and kelp-based things. It does all that just by an initial exposure to the VOCs in the seedling stage. It's a no-brainer. But it's a tech that we just had no idea. Only in the last maybe 10 years, and then this terpene thing, in cannabis, that's the entourage effect, and it really modifies how that cannabis is going to work. It's not so much THC, it's like that combination of THC, let's say, and other cannabinoids, which are also present, and the entourage effect of terpenes. And they can modify the effect that you feel, and more. I mean, they have multiple benefits and effects, and I've got that all listed, like which terpenes are we talking about, and what does science say they do, what do they do for people, what do they do for plants. So I got that all down, and I'm going to make a YouTube or some kind of presentation. Matter of fact, I'm going to be talking at the Future Cannabis Project, and a podcast coming up next month. Future Cannabis Project, if you want to look that up. The podcast information is already listed, the date and so forth. And one of the researchers that's worked at testing bioag products at the University of Kentucky, Steve Diver, is going to be in that podcast with me. So we're going to discuss biostimulants and then this new tech with volatile organic compounds of C. spherifernum strain TOC6. As it turns out, this same fungi, only this particular strain has that effect. See, so it's not like a standard thing within that particular genus and species. It's a strain, and this was found by accident, fortuitous accident. Just like we found another fungi that inhabits the root system of plants and increases yield in dryland crops and easy and cheap. It's called serendipity. It was found the same way, serendipity. Okay, it does so much, but see, the commercial agropesticide plant input industry is not interested because it's too cheap, it's too easy, too simple. It replaces a lot of stuff. It can really reduce the need for toxic genetic chemicals and crop rescue. We let the crop rescue itself. It doesn't need to rescue itself. It just activates the resistance, and it takes care of itself. And is just something that people made up? No, there's a lot of research. And right here, I mean, I got a 10-acre research farm with greenhouses, orchards, growth chambers. Yes, and I proved it. I'm not just talking about it. People don't necessarily like my style because, I just speak the truth. And maybe you don't like the story, but, that's what it is. And in this case, I'm saying that the effect we get from VOCs is similar to the effects we get from biostimulants. My company is one of the leading biostimulant companies. So I'm going against, that particular tech that I've been involved in for 60 some years. Okay, that's okay. If we find something better, yeah, of course. And I did. And it's a breakthrough. And it answers a lot of questions about how we can reduce pesticides, how we can and the benefits to humans. And the super benefit right now to the cannabis industry if they'd start using it, because I have the product available now. And we have some major cannabis growers on the West Coast who are starting to use it because the results are obvious. You can find out in 90 days. You know, I use auto-flowering feminized seed, and, you know, it's about a 90-day or so. And you find out. You can find out if it doesn't work. You know, you can call me a liar and get your money back. But that's the story. And so if you want to listen to the future cannabis project, and we're going to talk about it, and also my YouTube channel, Dr. Faust, or at Dr. Faust /Bioag. And then I'm going to have more stuff, YouTube, show the analysis reports that were done by Pre-Labs in Corvallis, Oregon. It's excellent, excellent data. We never had this ability before. It's giving me some answers I've been looking for. Like, how come some of that pot we had back in the day was so good, had a different effect, you know, and what we have now is very disappointing. Well, okay, now I know. now I know. Okay, terpenes. And so we know how to turn that around, you know. And it's pretty obvious too. I mean, if you actually try it, not just debate it and talk about it, you'll see what I'm talking about. It's already works and published. It's patented by the USDA. They did years worth of data. It's incredible data. I can show you the papers. We did 4 years and a whole range of plants, everything from tomatoes and vegetables, you name it. Now the USDA, their testing was exclusively tobacco. Yeah, tobacco seedlings and micropropagation, and they demonstrated the effect on tobacco. Okay, I'm not a fan of tobacco. Okay, and my best friends died because of it. So no. So I tried it on legal cannabis, hemp here in Oregon, and yeah, it's incredible. So there's no difference between the effect, let's say, between tobacco and cannabis, even though they're not related plants. Tobacco is related to tomatoes, yes, but it's really a cellular, molecular, and genetic pathway activation, or let's say molecular pathways are switched on. It's a switching agent, just like it switches things in your brain, these certain terpenes, if you ingest them. Good things, you know. So that's the story, and if you want to find out more, I got more, lots more. So tune in, turn on, and maybe drop out. Okay, this is Robert Faust, signing off from, I'll show you where we're signing off from. It's quite an interesting location here in Oregon. See, a lot of green, a lot of giant forests. And why is it so green and vibrant? Could be humus production too, couldn't it? Talk to you later.